How does vaccines work to prevent specific infections? Vaccine injections are given into your arm or in oral drops for your immune system to fight germs, the invaders, or we call them the antigens. Of course, vaccines are not given when you have a specific infection, but given when not having that infection. You are given a weaker or attenuated version of a virus or bacteria in the form of a vaccine. Vaccines contain weakened or inactive parts of a particular organism or the antigen that triggers an immune response within your body. Newer vaccines contain the blueprint for producing antigens rather than the antigen itself. Pfizer and Moderna vaccines are made using messenger RNA or mRNA technology that delivers a bit of genetic code to human cells to make the surface protein known as the spike on the SARS-2 virus. These proteins in your body activate the immune system, teaching it to see the spike protein as foreign and develop antibodies and other immunity weapons to fight. AstraZeneca vaccine, on the other hand, uses a chimpanzee adenovirus vector. This is a harmless, weakened adenovirus that usually causes the common cold in chimpanzee. It is generally changed so that it is impossible for it to grow in humans. The adenovirus vaccine vector, known as ChAd-OX1, was chosen as a suitable vaccine technology for the SARS-CoV vaccine, as it has been shown to generate a strong immune response from one dose in other vaccines. AstraZeneca has been proved to be safe and effective. When your immune system first encounters a vaccine, it activates two important types of white blood cells. First, the plasma B cells, the cells that make antibodies that are used to attack invading bacteria, viruses, and toxins. They also contribute to any allergic and autoimmune disease. The first antibody produced by the plasma cell is IgM, which form about 10% of all antibodies. Then there are the T cells, and they are tailored to identify a particular pathogen and kill it. These are also called memory cells, as they are primed up to face the virus because they have tested the vaccine given and being aware to identify the enemy. These cells have a long lifespan and they keep on lingering in your blood for decades. These cells are increased with the second booster shots. On the second exposure to the same vaccine or the germ, the B cells that remain from before can rapidly divide and create increasing numbers, leading to a second spike in the number of antibodies circulating. The second dose also starts the process of B cell maturation and binds to a particular pathogen. Memory T cells, meanwhile, also proliferate rapidly and give long-lasting immunity. It is observed in the case of COVID vaccines, the immunized person against the COVID-19 would lose approximately half of their defensive antibodies every 108 days or so. As a result, vaccines that initially offered, say, 90% protection might be 70% effective after 6 months, or 7 months, according to Miles Davenport of Kirby Institute, University of New South Wales, and his colleagues. Immunological studies have shown that there is a decline of antibody levels among vaccinated individuals who are prone to having the risk of breakthrough infections. Those individuals who are vaccinated can still catch COVID-19, but you are most likely to experience only mild symptoms. Moderna joined Pfizer by revealing new data showing the waning impact of its COVID-19 vaccine, requiring the need for a third booster shot. In the US, it has been recorded that 88 breakthrough cases among 11,431 individuals who had been vaccinated between December 2020 and March of this year. Now let's discuss about the long-lasting antibodies. It is observed that there are positive signs of long-lasting antibodies for COVID-19 infection. People who recovered from mild COVID-19 have bone marrow cells that can churn out antibodies for decades, although viral variants could dampen some of the protection they offer. So it is found that people who have had the infected SARS-CoV-2 will probably make antibodies against the virus for most of their lives, as the researchers have found long-lived antibody-producing cells in the bone marrow of people who have recovered from COVID-19. According to Menno van Zelm, an immunologist at Monash University, Melbourne, a study provides evidence that immunity triggered by SARS-CoV-2 infection will be long-lasting, and the vaccines will also have the same durable effect. The effect is observed due to the memory cells patrolling in the blood expecting a reinfection, and the bone marrow plasma cells hiding in the bone marrow keep tricking out antibodies for decades. A plasma cell is our life history in terms of the pathogens we have been exposed to, says Ali El-Abedi, a B-cell immunologist at Washington University in St. Louis, Missouri, who led the study published in Nature on 24th May this year. Researchers presume that SARS-CoV-2 infections triggers the development of bone marrow plasma cells in mild infections of COVID. But in severe cases of COVID-19, there is a disruption of the plasma cell formation. El-Abedi's team states that the ability of some emerging SARS-CoV-2 variants to blunt the protective effects of antibodies means that additional immunizations may be needed to restore levels, meaning a third booster may be needed. Hope this video talk was useful. Stay safe at home and be ready for your third booster jab. Goodbye for now.